Chapter 2, Chromosomes and Cellular Reproduction, including mitosis and meiosis. It's expected that you still remember something about cell structure and function in terms of the basic structures of a prokaryotic cell and the basic features of a prokaryotic cell, as shown on this slide. And for the eukaryotic cell, again, the basic structures, basic functions, and of those organelles and basic characteristics of eukaryotic cells. And that's because we're going to build on that knowledge. So I'm going to talk a little bit about genetic characteristics of prokaryotes in this chapter, and then we will spend one entire chapter dedicated to the genetics of prokaryotes. So as you can see, the domains archaea and eubacteria are both the prokaryotic domains, and those contain organisms that have no nuclear envelope. That's why they're called prokaryotes. And their cell structure is similar in a lot of ways. They're fairly simple. They have DNA, ribosomes, cell wall, cell membrane. But it turns out that some of the genetic processes in the archaeans are more similar to the eukaryons than the eubacterians. And it turns out that, because that's true, or as a result of that, we have been able to figure out that eukaryotes probably evolved from an archaean that engulfed or took in some eubacteria and started to live in a symbiotic relationship together. That's how we think the very first eukaryotes formed billions of years ago. And so it makes sense that some things in the archaeans are similar to our modern day eukaryotes. And two of the things, just in very general terms, some of the details of transcription are more similar between the archaeans and the eukaryotes. And the archaean DNA is wound up with proteins called histones, which eukaryotic DNA is also wound up with histones. So that's kind of interesting. Eubacterians do not have histone proteins wrapped with their DNA. However, all the prokaryotes typically have a single circular chromosome. And we say usually, there are some exceptions, but usually a single chromosome, and then multiple smaller circular pieces of DNA that we call plasmids. However, most of our discussions in this course are going to circle around eukaryotes. And eukaryotes have a nuclear envelope surrounding their DNA. They typically have many linear chromosomes, not just one. And they have their DNA wrapped around histone proteins in a very highly and tightly wound arrangement. However, eukaryotes do have DNA in the mitochondria and chloroplasts, and that DNA is circular, reminiscent of the origins of those organelles coming from ancient eubacterians. And then you have the viruses. The viruses are not organisms. They're not considered alive because they cannot replicate on their own. But it is important to discuss viruses. And viruses, the structure of a virus is typically a protein coat. They may also have a phospholipid coat, but they all have at least a protein coat and then some kind of nucleic acid on the inside, which we do typically call a chromosome. So the viral structure is some kind of coat, minimally at least a protein coat and sometimes an additional lipid coat, and then on the very inside, the genetic material. And viruses are very interesting. They are actually best studied in parallel with the genetics of their host cells because they evolve together, they co-evolve. And we'll talk a little bit about viruses in the course here and there. So now we get to some very critical terminology for this section of the course, and maybe for the whole course. Because if we're going to talk about cell division, especially cell division in eukaryotes, we're going to use some terminology that seems simple at face value, but actually can be a little tricky or a little complicated. And the question that I have here, the first question is, what do we mean when we refer to a set of chromosomes? And then what is meant by the terms such as diploid, monoploid, haploid? So these are important questions. I have a whole special tutorial or a couple of tutorials on this. So I'm going to explain it briefly here, and then I will expect you to watch those other tutorials and try the practice problems and see if you're getting stuck or if you're understanding the concepts. So a set of chromosomes. A set of chromosomes, in this picture we have a photograph, an electron micrograph. Well, it might be a light microscope photo, but they've colorized the chromosomes with a fluorescent stain, and they've organized them artificially into pairs. And pairs, like a pair of shoes, is 2. So humans have 23 pairs of chromosomes in each cell, but that means there are 46 total chromosomes, because 23 pairs is 23 times 2. So in the picture you see this colorized image of the 46 chromosomes that would be in a human cell, and they've organized them according to the way that we name the chromosomes, which we use numbers to name them. So you have 2 copies of chromosome 1 and 2 of chromosome 2 and 2 of chromosome 3 and 2 of chromosome 4, etc., etc. So 46. They're not all the same size, and some of them appear to be fragmented, but that's really just because of the way the stain didn't quite stick to all parts of each of the chromosomes. But in any case, a set of chromosomes, a set of anything, would be one of each different type. So 1 copy of chromosome 1 and 1 copy of chromosome 2 and 1 copy of chromosome 3 and 1 copy of chromosome 4, etc., all the way up to chromosome 22 and then 1 of the X chromosomes in this image. All of those would make up 1 full set. The other set, which is also in this cell, would be made up of the other copy of chromosome 1 and the other copy of chromosome 2 and the other copy of chromosome 3 and the other copy of chromosome 4, etc., etc., all the way up to the other copy of X. That would be the second set. So you have 2 full sets. In this image, they're not organized in their sets, they're organized in pairs. So the 2 copies of chromosome 1 are together. The 2 copies of chromosome 2 are together. But there are 2 sets total in this image, in the entire image. So the terms diploid, monoploid, and haploid, what you need to remember is that ploid, P-L-O-I-D, means set or sets. And then whatever prefix is in front of that tells you how many sets. So diploid would be 2 sets. Monoploid would be 1 set. And those terms refer to, they are adjectives and they describe a cell. So diploid describes a cell. A diploid cell contains 2 sets of chromosomes. A monoploid cell contains 1 set of chromosomes. Now, how many that is? In humans, a set is 23 chromosomes. But in a mouse, a set is 20 chromosomes. So the number of chromosomes that are in a set, that depends on what species of organism you're referring to. But a set is always one of each different kind. So in this photo, one of each different color chromosome makes up 1 set, and then the other one of each different color makes up the other set. So if a cell contains 2 sets, then it would also contain 2 copies of each particular kind of chromosome. A monoploid cell would only contain 1 set. So 1 copy of chromosome 1, 1 copy of chromosome 2, 1 copy of chromosome 3, etc. And there are monoploid cells in the human body. We call them gametes, eggs or sperm. So most of the cells in the human body are diploid, and a smaller number are monoploid. So then you get this word haploid. Haploid is, technically, it means half of whatever is considered the normal ploidy. So in humans, diploid is the normal. In most organisms, diploid is the normal. And so haploid is half of that. So if 2 sets is what cells normally contain, then half would be 1 set, half of the ploidy. So haploid isn't an exact number of sets, it just means half of whatever is normal. Most of the time, since most organisms are diploid, haploid does mean 1 set of chromosomes. So most of the time haploid is synonymous with monoploid. But there are organisms whose normal ploidy is not diploid. So let's say you had an organism who had its normal ploidy was tetraploid, 4 sets. Cells normally contain 4 sets of chromosomes. Then in that case, haploid for that organism would be 2 sets. So haploid in that case would be equivalent to diploid if tetraploid, 4 sets, was the normal case, normal situation. So gametes, sex cells, are haploid, and they're also often monoploid because most organisms are diploid. But if the organism is not diploid, then haploid can mean some other number of sets. It just means half of whatever the normal ploidy of the organism is. Now there are parts of chromosomes. One part is called the centromere, and that's where the spindle fibers will attach during cell division. There's a little protein apparatus called the kinetochore that attaches as well right at the centromere. The centromere is composed of DNA. The kinetochore is some proteins that attach to the DNA. And then at the ends of the chromosomes, we call those the telomeres. Now, on the left side of the screen, you see a monad chromosome. So monad chromosomes that's the form of the chromosome that exists, for example, in the cell cycle in G1. Then during S phase of the cell cycle, the chromosome is replicated and it becomes a dyad, which still counts as one chromosome, but we call it the dyad form, which is in the center of the screen. So on the far left is a monad chromosome. That counts as one chromosome. In the middle of the screen here you see a dyad. A dyad still counts as one chromosome, but in our mind we remember it's made up of two sister chromatids. But when the two chromatids are connected at the centromere, we call it one dyad chromosome. So you have to be very aware of how the language is used in genetics. It will help you quite a bit. Eventually, when the dyad chromosomes are pulled apart, that would be during anaphase of the cell cycle, or it would be anaphase 2 of meiosis. When dyads are pulled apart, at that point they become separate monad chromosomes. But when the two chromatids are together, we call them chromatids because when they're together, the whole thing is a chromosome. So a dyad chromosome is made up of two sister chromatids. A monad chromosome is the form that you see on the left. So certain phases of cell division always have certain chromosome forms, and you will have to memorize which phase of the cell division has which form of the chromosomes. Now, one way to identify and classify chromosomes, when you're looking, if you're a cytogeneticist and you're looking at, for example, an image that's shown here would be what you would see under the microscope, you can identify the chromosomes partially based on how the centromere is positioned. They also measure the arms of the chromosome, but partially you can do it based on how the centromere is positioned. And so we have names for centromere positions. One is metacentric. Metacentric is the second one on the list. It's where the centromere is exactly in the middle of the dyad chromosome. Submetacentric is at the top. That's where the centromere is slightly towards one direction more than the other. Acrocentric is at the bottom. That's where the centromere is very much towards one end of the chromosome. And telocentric is where the centromere is virtually at the telomere. In humans, we do not have any telocentric chromosomes, but other organisms do. In humans, we have metacentric, we have submetacentric, and we have acrocentric chromosomes. Now, centromeres also have the same position if they're on a monad chromosome or if they're on a dyad. It's just that the photograph of the chromosomes is always done in metaphase, and metaphase cells always have dyad chromosomes. So when you see a picture of chromosomes, you will always see dyads because the pictures are always done in metaphase for logistical reasons. It's just easier to photograph them at that time. Cell cycle, I have a tutorial. I assume that you know most of this and you just can review and refresh, but there's a tutorial for you to watch on the cell cycle. So make sure you're familiar with this. But let's get back to our counting chromosomes. All right, because this is how I will be able to figure out if you really understand what's happening at every stage of the cell cycle and then later at every stage of meiosis. So this is the cell cycle, G1S, G2, and then M phase, mitotic phase, consists of prophase / prometaphase, metaphase, anaphase, and telophase. So I want you to pay attention to what's at the bottom of this slide. The number of chromosomes per cell, you start off in G1 with 4, in this image, 4 monad chromosomes. Nothing changes in terms of the number of chromosomes, even when the chromosomes become dyads. So replication during S phase does not change the number of chromosomes per cell. But the number of DNA molecules, in other words, another way of looking at it is the weight of the DNA in the cell, doubles during S phase because monad chromosomes become dyads with 2 chromatids, and so the weight doubles at S phase, but the number of chromosomes does not change at S phase. So it's 2 different ways of looking at what's happening in the cell. When you get to, so then whatever number you have there stays the same. And then in anaphase, you'll notice that the number of chromosomes per cell doubles because you go from having 4 dyad chromosomes in metaphase, and now in anaphase, you have 8 monad chromosomes in that cell. But the weight of the DNA doesn't change. You have 8, and you still have 8 when you go from metaphase to anaphase. But both of the numbers are split in half when you go from anaphase to telophase. And remember, they're comparing one cell in anaphase to one of the product cells in telophase. So this cell in anaphase has 8 chromosomes and 8 molecules of DNA. And then that number, each one telophase cell has 4 chromosomes and half as much of the DNA as was in anaphase. So it's very interesting how those 2 numbers change in different places depending on whether you're looking at the counting of the actual chromosomes or the weight of the DNA in the cell. So several ways I can ask questions, and like I said, I've given you some practice problems in this module. First of all, just by looking at, and remember, if we're talking about cell cycle, and I give you a picture like this, and I ask what phase of the cell cycle is depicted here, you should be able to identify from a picture like this, a diagram, that this has to be anaphase because the chromatids are being pulled apart. The dyads are now pulled apart into monads. How many chromosomes does this cell have right now? You count each one individually. Monads, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. How many chromosomes did this cell have in G1? So if it has 12 chromosomes in anaphase, but remember the number of chromosomes doubles in anaphase because you're pulling apart dyads. The original cell had 6, and in G1 it would have had 6 monads. So you kind of have to be able to think forwards and backwards through this. So the answer would be 6. And in G1 those would be monads. If this cell's DNA weighs 10 picograms right now, how much did this cell's DNA weigh when the cell was in G1? So if G1 was 6 monads and this is 12 monads in this picture, then you would divide it by 2, so 5 picograms for G1. In G2, the chromosomes are replicated. So in G2, it would also be 10 picograms. So G1 would be 5, and G2 would be 10 picograms. So I can ask you about the chromosome count, and I can also ask you about the DNA weight. And those are 2 different things, although they require some of the same logic and some of the same understanding of what's happening in each phase of the cell cycle.